Today we are looking forward to an exciting presentation by Vera Ehrenstein and Mohamed Abouraghi from our Real4Reg project, from our consortium, followed by a roundtable discussion with different stakeholders from the regulatory, academic, and also patient's perspective. And now I would like to hand over to Claudia from INFARMED.
Good morning, everyone, and on behalf of the Real4Reg Consortium and INFARMED, welcome to this Real4Reg workshop. Just a few housekeeping rules. Let me just share the slide. The workshop is being recorded, as you noticed. During this workshop, please keep your microphone muted when you are not speaking. And if you have any questions or comments during the workshop, kindly use the chat to ask questions and provide the comments. As Britta already mentioned, this workshop is about lessons learned on exploring real-world data in relation with Real4Reg project. And this workshop focuses on the description of the study population for ALS and breast cancer. We will start with a presentation with first results and challenges in OMOP implementation by Vera Ehrenstein and Mohamed Abouraghi, followed by a panel discussion. I will present briefly Vera and Mohamed. Vera is the scientific lead of Work Package 1 in Real4Reg project. She's a professor for Phase 4 studies at Department of Clinical Epidemiology in Aarhus University, Denmark. She has served as scientific coordinator of several large multinational multi-database studies on safety of medicines and vaccines. Multi-database studies, including regulatory mandated and industry commissioned studies. She has experience working with major European databases, including Nordic registries and databases in the Netherlands, Italy, Spain, United Kingdom, Hungary, France, Germany, the United States, and most recently, Portugal. She has served on the board of directors of the International Society for Pharmacoepidemiology and is currently a member of the steering group of the European Network of Centers for Pharmacoepidemiology and Pharmacovigilance, ENCEP. In that capacity, she has continually contributed to the development of methodological guidelines, most recently as the coordinator author of the guideline for conducting safety studies in pregnancy and breastfeeding using real-world data. Mohamed Abouraghi is a researcher and PhD candidate at Fraunhofer SCHI. His work focuses on biomedical data science and artificial intelligence, particularly in the context of complex and neurodegenerative diseases. Mohamed has worked with large-scale data sets such as UK, Biobank, and PPMI, and is currently actively involved in the Real4Reg project. He has a background in pharmacy and a master's degree in life science informatics from BIT in Bonn. I will now hand over to Vera to start the presentation. Thank you.
Thank you very much, Claudia, for the nice introduction. Can you please confirm that you can see my slide in the presentation mode?
Yes, we can see.
Thank you very much. So good afternoon, everyone. Thank you very much for attending this workshop where we present the initial results of use case 1 in the Real4Reg project. I would like to gratefully acknowledge my co-leads of the Work Package 2, Associate Professor Elizabeth Horvat-Puho and Statistician Istvan Bakos, and also all the collaborating institutions and colleagues who have contributed with their hard work and expertise into this presentation. So, let's see if I can advance. No, yes. Taking one step back and presenting the Real4Reg project as a whole, the Real4Reg aims to develop, optimize, and implement artificial intelligence methods for real-world data analysis in regulatory decision-making and health technology assessment along the product lifecycle. This project is 4 years long and has started in 2023. And we just hit the finish stretch. We're about to complete this project at the end of this calendar year. This is a large scientific consortium consisting of 10 partners from 6 European countries and it is led by BfArM, whose leadership I also gratefully acknowledge. So just to give you a brief overview of the databases that contribute to the scientific results of Real4Reg. They are presented in this table. The data in Real4Reg come from 4 different countries, from Denmark and Finland, representing Nordic registry data, from Germany, representing claims data, and from Portugal, also representing a registry type data. All these data are accruing on individual level in the automated secondary national regional electronic health records database. And this is an example of the real world data that are increasingly used in both post-authorization and pre-authorization space. The overarching objectives of the Real4Reg project is to develop new and optimized methods for analyzing real-world data, including addressing the challenges and taking advantage of opportunities across different healthcare systems. Ultimately, we hope that this will support better decision-making about medicines and other treatments, and ultimately benefits patients and public health. And here you can see here some of the specific objectives of Real4Reg is to enable the use of real-world data in real-world evidence to establish or better establish its value in the society, and to impact how real-world data are used and analyzed. And this project aims to provide solutions to those objectives. There are 4 use cases, scientific use cases, comprising Real4Reg. So the aim again is address the need to demonstrate the potential of real-world evidence in regulatory decision-making and health technology assessment. And in that, Real4Reg is employing these highly relevant use cases, some on the disease side and some on the medication use side. So use case 1 and use case 2 are addressing the pre-authorization and evaluation side of the product lifecycle, and use case 3 and 4, which are not focus of today's presentation, are addressing the post-authorization side. So let's delve into the main topic of today, use case 2. Let me talk a little bit about the background and the rationale of use case 1. As some of you who are professionals in this area, they have probably seen this sentence many times in the papers saying that randomized controlled trials do remain the primary requirement for the initial authorization of treatments based on their efficacy and initial or short-term safety. Real-world evidence, and under that we understand evidence generated from real-world data, has been traditionally and already for a long time used for assessing post-authorization or long-term safety and effectiveness and also for effectiveness monitoring of treatment. However, regulators, including the European regulator, the European Medicines Agency, have started to recognize the potential of real-world data, so data accruing outside randomized trials, to complement data from randomized controlled trials in regulatory decision making, including not only post-authorization, but also pre-authorization phases. And the value of real-world data is especially potentially high in areas where randomized controlled trials have limited applications, and that applies to rare diseases, severe conditions, vulnerable patient subgroups, and situations where there is lack of clinical equipoise, as traditionally these types of situations are not part of randomized clinical trial populations for a good reason, because exclusion of those increases internal validity of randomized clinical trial results. We have also, those of us who work in this area, have also been aware of some of the barriers that stand in the way of use of real-world evidence in the pre-authorization and evaluation phase. Some of them are listed here. For one, there is lack of awareness and knowledge among not only the public, but also in some professionals about the available data sources. Number two, among those who know something about the data sources, there is some skepticism, sometimes justified, and concerns about data quality and validity. And those researchers who work every day with their hands with those data are really acutely aware about sometimes or often slow data access for studies, even if the data are collected for regulatory and health technology assessment purposes. So, as I mentioned before, use case 1 is one of the use cases in work package 1. And here we start by a very relatively simple analysis. We would like to conduct a basic epidemiologic study where we describe, using real-world data in several countries, populations of patients with amyotrophic lateral sclerosis, ALS, and breast cancer. And these types of studies are often conducted in our world to understand the clinical course and the prognosis of the diseases in the population. Why did we select ALS and breast cancer as kind of to showcase the use of real data in pre-authorization stage? There are several reasons. Number one, these two diseases represent two diverse, or I should say different, pre-authorization settings in which real-world evidence use may be especially relevant. Breast cancer is the most common and a very well-studied malignancy in women. In women, breast cancer has well-developed treatment protocols. It is also known that prognosis of breast cancer differs by patient characteristics. Specifically, less is known about breast cancer in men, although it does occur. And because it is very rare, breast cancer is very rare in men, men with breast cancer represent an understudied small subgroup with a less optimal prognosis than do women. ALS, on the other hand, is a very rare motor neuron disease characterized by high fatality and absence of cure. Currently, pharmacotherapy with riluzole is the only approved option used for symptom relief among patients with ALS. So to summarize, because evidence from randomized control trials in rare subgroups of patients with breast cancer and also in ALS patients is difficult to obtain. It is difficult to obtain because the populations in those subgroups and those patient groups are small. These patient populations potentially may stand to benefit from the use of real-world data in the pre-authorization stage. Here are the specific objectives of use case 1. We start from the beginning. We start with having actual basic data access, pre-processing, including data management as an objective of this study. So we would like to learn about what it takes to access data in the 4 participating countries. We would like to carry out the pre-processing tasks, including quality control. And as you will see later in this presentation, we would like to convert the diverse data from the different databases into a standardized common data model called OMOP for the analysis. Then we would like to provide a descriptive overview of breast cancer and ALS populations, including pre-morbid characteristics, so pre-disease characteristics, characteristics of the disease itself. We would like to look at treatments, and finally, we would like to look at prognosis. Now, I would like to stress that we are not concerned here about the clinical or scientific conclusions from these analyses. What we would like to do, we would like to conduct a proof of concept study. We would like to compare results that we obtain against the published evidence and to create the best practice guidelines as a result of that. So when we obtain results on the same objectives from different heterogeneous data sources, we would like to examine on the sources of heterogeneity of the results across the country-specific patient populations and across different data types, across different coding practices, accessibility, representativeness, completeness, temporal variation, and validity. So in terms of methods, we have conducted a cohort study, and this very simple graph represents the study design. So for breast cancer, we identify in the routinely collected data in the databases incident breast cancers, and we say that the date on which breast cancer was diagnosed was the index date. We assess eligibility criteria 5 years before the index date, and then we follow patients forward. And the end of the follow-up, shown here on the right, depended in this study on the specific database, depended on the data refresh status at the time that the data were extracted. So this represents the study design for the breast cancer population, but it is pretty much the same for the ALS population. So we're doing basically 2 cohort studies here. So in terms of data access and management, as the first step, we always need to get ethical or other governance, follow other governance procedures to be able to legally obtain and analyze the data. So we have filled out protocol-based data applications in each country's data custodian. So we are not the owners of the data. We apply to data custodians to analyze these data on a protocol basis. Upon receiving access or receiving data from data custodians, we've been conducting local data checking to ensure that the data that we have requested have in fact been delivered. And those who work in this area know that more often than not, something actually may be missing from what you have requested. So it is very important to check that the delivered data or the access data are the data that have been requested. And then we do data management that consists of the routine cleaning and plausibility checks. And the next step would be of data access and management would be coding the data according to the protocol, according to operational definitions of study variables. And as you can imagine, since we have 4 countries in this project, each data has its own flavor, meaning that the coding practices and some of the coding conventions and classifications differ across the different databases. So I'm going to pause here, and I'm going to give the floor to Mohammed, who will talk a little bit about this step of converting the native data to the OMO format. And then I will take over again and talk about the results of use case 1.
Thank you very much, Vera. For the current presentation, just a quick question. Do I need to share the slides, or can you maybe navigate them for me? It would be much.
I'd be happy to advance if you like.
Thank you very much.
And then I need to share, I guess, right? Okay, here you are.
Yep. Thank you very much. So yeah, I will start by briefly setting the scene for how we actually bring very heterogeneous national data into a common data structure. And what you see here is the standard extract, transform, load workflow, or the ETL, which is the backbone of any OMOP implementation. And we start with the national medical databases from different countries, such as Finland, Denmark, and Portugal, and Germany. And these data sources differ substantially in the structure, in the coding systems, and at the level of clinical detail. The first step is extraction, where we retrieve the relevant tables from the source systems and place them into a staging area. And at this stage, the data is still very close to their original country-specific formats. The core of the process is the transformation step. And this is where most of the work happens, basically. We clean the data, we normalize the formats, such as the dates and the units, and crucially map local coding systems to standardized vocabularies that are used by the OMOP common data model. And after that, the transformed data are loaded into the OMOP common data model, which gives us a harmonized analysis-ready representation across all countries. Once the data are in the OMOP common data model, we can use a shared analytical layer, common tools, common definitions, and reproducible methods, while still respecting that the data originate from very different healthcare systems. I think we can move to the next slide. Initially, the ETL design was developed based on the metadata that was available to us upfront. So the table descriptions, the variable lists, and the documentation that was provided before we had direct access to the data. However, once we obtain the data access to the real data sets, we quickly see that this initial design has to be adapted a little bit. And in practice, we encounter different variable names, unexpected table structures, and in some cases, information that was documented but implemented differently in the actual databases. Another important adjustment was at the technical level, where due to the size of some national data sets, we had to switch to an in-process database approach rather than keeping everything in memory, simply to make the ETL scalable and robust. The key point here is that the ETL development was not a one-off task. It required iterative adaptation of scripts as we moved from documentation to real-world data. And the main learning from this experience is that critical importance of early access to data and having accurate, detailed metadata available. Even small discrepancies at this stage can have downstream effects on harmonization and analysis, and this is especially relevant in a multi-partner setting like Real4Reg, where aligning the expectations early can save substantial effort later on. Next slide, please. So, building on the ETL process I just described, I now want to focus on one of its most critical components, which is the target schema mapping. The central challenge here is that each country uses its own coding systems, its own data structures and conventions. So on the left here, for example, you see examples from Finland, Denmark, and Portugal, each with its own way of representing the same underlying clinical concepts. And then the target schema mapping is the step where we translate these country-specific codes and structures into a standardized vocabulary and schema, as defined by the OMOP common data model. This mapping is essential because it allows us to align otherwise disparate data sources and to analyze them cohesively within a single framework, rather than treating each country as a separate special case. In practice, this means that mapping local diagnosis, procedure, and drug codes to OMOP standard concepts and assigning them to consistent tables such as, for example, the person, the condition, and the observation tables in the OMOP common data model and associated date fields. What's important to emphasize here is that this is not a purely mechanical lookup. Many concepts do not map to one-to-one concepts, and decisions must be made about granularity, clinical meaning, and how much information can be preserved without introducing bias. And once this mapping is done, the OMOP schema gives us a shared analytical language, the same cohort definitions, outcome definitions, and analysis code that can then be applied across all the partners. Next slide, please. Yes, once we define the target schema, the next question is, how can we actually map local medical codes to OMOP concepts in a consistent way? For this, we rely on Athena, which is a searchable vocabulary database that is maintained by the Odyssey community. And it serves as a central reference point that brings together many different clinical vocabularies and links them to the OMOP standard concepts. In practice, Athena helps us to identify a local code, whether it is a diagnosis or a procedure or a drug, and then find its corresponding OMOP concept together with additional contextual information. So the main three elements that are important to us here: first, the domain ID, which tells us whether this concept is a condition, a drug, a procedure, observation, or other clinical domain. And then the second is the concept ID, which are unique identifiers that allow us to represent the same medical concept consistently across all datasets and countries. And third, the hierarchical relationships, which define the parent-child links between the concepts and allows us to work at different levels of clinical granularity when needed. Together, these features make the vocabulary mapping more transparent and reproducible, and they are essential for ensuring that the analysis performed on OMOP data are comparable across the partners. Next slide, please. So to make this more concrete, this is an example of vocabulary mapping using Athena. So here we have an example concept, which is gestational diabetes mellitus. In OMOP, this condition is represented as a standard concept with a unique concept ID, for example, the number that is in front of you here, and it is derived from the SNOMED vocabulary and classified as a disorder. By assigning this concept to the condition domain, we define where it belongs in the OMOP schema and how it will be treated in downstream analysis. And what is particularly important here are the hierarchical relationships, which you can see visualized on the right. For example, gestational diabetes has a parent concept such as diabetes mellitus in pregnancy, which represents a broader clinical category. At the same time, it can have child concepts that capture more specific situations, such as different diagnostic formulations or subtypes recorded in a local coding system. This hierarchy gives us analytical flexibility. If we want a very specific cohort, we can focus only on the exact gestational diabetes concept. Or if we want a broader cohort, then we can include the parent concept and automatically capture the related pregnancy-associated diabetes conditions across all datasets. The key advantage is that these decisions are made once at the concept level and then applied consistently across all the countries, regardless of how the condition was originally coded locally. We move to the next slide. Yes, while Athena is very powerful, there are some challenges that we encountered when using this in practice. So one category of challenges relates to the missing specific codes. Some country-specific codes, such as special reimbursement codes or socioeconomic indicators, simply do not exist in Athena because they are not part of international clinical vocabularies. And a concrete example is shown on the right here, where certain chemotherapy treatment regimens or country-specific combinations are recorded as non-standard. And in these cases, we relied on specialized vocabularies such as the hem-onc or the hematology oncology vocabulary. But even then, not all local formulations could be mapped cleanly to a single standard concept. The second, and in some cases more fundamental challenge, is missing entire vocabularies. For example, extended national classifications like the Danish SKS classification system is not fully represented in Athena. And this means that some local codes have no direct equivalent in OMOP and cannot be mapped one-to-one. The example at the bottom illustrates this. So highly specific Danish SKS code had to be simplified to a more general ICD-10 concept, and such as mapping the several subcodes of G12.2 back to the base ICD-10 category. As a result, the main learnings here that manual mappings were unavoidable. There will be some loss of specificity that is necessary, and that mapping certain highly unique local codes was simply not feasible. And these trade-offs are important to acknowledge because they directly affect the data granularity and must be considered when interpreting downstream analysis. Slide, please. So beyond vocabulary mapping, another major challenge we encounter relates to the missing information in the source data itself. So the common data model is intentionally very detailed, and it assumes that a wide range of information is available and explicitly recorded in the source system. But in reality, this assumption does not always hold. In several datasets, we observe mixtures of structured data and free text. For example, some information such as the number of dispensed drugs may be embedded in text fields rather than stored in a structured format. And extracting this information often requires a manual review or custom parsing, which is feasible only for selected subsets of the data and not at a scale. We also encountered cases where key OMAP fields were not simply recorded consistently in the source data. A typical example, for example, is drug exposure information, where variables were such as drug exposure end date or days of supply were missing or incompletely captured and had to be derived. These gaps limit our availability or ability to represent treatment duration accurately and can directly affect the downstream analysis again, such as exposure definitions or time at risk calculations. The main learning is that the importance of standardized input formats and the need for complete structured information, especially for drug administration. And where this information is missing, assumptions or simplifications become unavoidable, and these need to be clearly documented and considered when interpreting results. Next slide, please. Yes, it's also important to emphasize that introducing standards does not automatically remove the ambiguity. So a good example of this is the ATC classification system, which many of you may already be familiar with. So ATC, or anatomical therapeutic chemical classification, organizes the drugs based on their anatomical, therapeutical, and chemical properties and assigns a structured code to each drug or group of drugs. As shown on the right, ATC is hierarchical with multiple levels, and it ranges from broad anatomical groups down to specific chemical substances. For example, if we take metformin as an example, we can represent it at a different level. For example, at a higher level, it belongs to the alimentary tract and metabolism group, than to drugs that are used in diabetes. And only at the most specific level does it represent metformin as a chemical substance. The ambiguity rises because different analytical questions require different levels of this hierarchy. If you map drug exposures at a very broad ATC level, we may group together drugs with substantially different mechanisms of actions. On the other hand, if we map everything at the most specific level, we risk the fragmentation and the reduced statistical power, especially in smaller data sets. So while ATC provides a standardized structure, the choice of mapping level is still a methodological decision and not a purely technical one. Next slide, please. Another important example here is the ambiguity of standard vocabularies, for example, that comes from RxNorm, which we use extensively for drug mapping in OMAP. So RxNorm provides a standardized nomenclature for clinical drugs, offering normalized names and unique identifiers, and linking medications across different drug classification systems. And if we compare it to ATC, it is much more granular and explicitly models relationships between ingredients, strengths, dose forms, and branded products. And you can see the diagram on the right. So it illustrates this complexity. A single active ingredient here, for example, cetirizine, can be connected to multiple clinical drug forms or multiple strengths or multiple brand names, and they're all linked through different relationship types. This richness is extremely valuable, but it also introduces ambiguity at the mapping stage. For example, if we map a local drug code, we have to decide whether we want to represent it at an ingredient level or a clinical drug level or the branded product level. And each of these choices would answer a different research question. Ingredient level mappings, for example, support class-based analysis, while branded or dose-specific mappings are more appropriate for detailed pharmacological studies. Also in the Nordic context, this is further complicated by country-specific identifiers such as the VMR or the Nordic article number, which was linked to the standard RxNorm concepts by FIN-OMAP. And while this linkage is extremely helpful, it still requires careful validation to ensure that the intended level of detail is preserved consistently across the data sets. And next slide. So this slide brings together the previous two examples and highlights one of the most challenging situations that we encounter, which is the one-to-many mappings between standard vocabularies. So in this case, the issue arises when a single ATC code represents a combination of products rather than a single active ingredient. And when we translate this into RX norm, which explicitly models ingredients that the single ATC code naturally maps to multiple RX norm concepts. And in this example, for sitagliptin and simvastatin. From a technical perspective, this is expected and correct. However, from an analytical perspective, it may introduce important decisions. So we treat, for example, the question is, do we treat this exposure as a combination therapy, or do we split it into its individual components, or do we define a higher level exposure group? And each of these choices would lead to a different cohort definition. What's crucial is that these are not errors in mapping, but inherent ambiguities that arise when we bridge vocabularies designed for different purposes. Next slide, please. Yes, this slide also highlights a slightly different but equally important issue. So this time between ATC codes and VNRs. In principle, the VNR is expected to be unique identifier for specific medicinal products. However, in practice, we found some exceptions. So in some cases, the same VNR is linked to multiple ATC codes, often because the product was approved for multiple indications or existed in slightly different formulations. And the diagram here illustrates the situation where we have two ATC codes that both point to the same VNR, and this challenges the assumption of a one-to-one mapping. When such mappings are translated into OMOP, particularly via RX norm, this can lead to an inflation of drug exposure counts, simply due to how concepts are represented. And this is simply a consequence of how different classification systems are designed, again, for different purposes. The key learning at this point was that relying solely on standard concepts can sometimes be misleading in these cases, and instead manual verification of mappings becomes necessary, or alternatively, the direct use of source concept identifiers such as ATC or VNR may be more appropriate for certain analyses. Next slide, please. Yeah, so far I've shown different sources of ambiguity in standard mappings, and here we summarize their practical impact on the OMOP data itself. So the core problem is what we refer to as data inflation, and in some cases, a single health event in the native source data represented as one row can generate multiple rows once it is transformed into the OMOP format. And this typically happens when one source code maps to multiple standard concepts. So for example, on the left here, you see a simplified representation of a native record where a reference ID, a vocabulary code, a date, and a person ID together represent a single clinical event. And then after transformation, the same event appears as several entries in the OMOP condition occurrence table, with each with its own unique condition occurrence ID. And the challenge is that the standard OMOP schema does not provide a built-in mechanism to preserve the explicit linkage between these related rows. And as a result, it becomes a little bit difficult to reliably identify unique health events, especially if analyses implicitly assume that each row corresponds to an independent occurrence. If this is not handled carefully, it can lead to the inflated event counts. And this is why mapping decisions and how we operationalize them in ETL pipelines is not just a technical detail, but we do have direct methodological consequences. Next slide, please. So now I will show briefly how we address this issue in practice. So the core idea of the solution is to separate the standardization from traceability. And while OMOP requires that each record has a unique occurrence ID, we introduced an additional identifier that preserves the link back to the original source event. And concretely for each native health event, we generate a stable source level identifier, such as a unique ID before transformation. And when this event maps to multiple OMOP rows, each of those rows receive its own condition occurrence ID as required by OMOP, but they all remain linked to the same source identifier. And this linkage is stored in a separate linking table, which explicitly connects the source reference ID with the corresponding OMOP occurrence ID. This approach allowed us to accurately track which OMOP pros originate from the same underlying health event during the analysis. And as a result, we can avoid double counting or reconstruct unique events when needed and maintain the transparency about how mappings were performed. Important.
We need to accelerate a little bit, Madam Vera. Thank you.
Sorry?
We need to accelerate a little bit.
Yeah, okay. So I have just one last thing, is that the main learning is that the standard vocabularies and OMOP common data model is essential to harmonize the data across countries. And what matters ultimately is the ability to preserve the link between the standardized records and the original source events. And the last slide, we're just talk about the computational aspects. So this is the final slide. Depending on the population size and the observation period, the source data sets can become very large. And in secure data environments, this is often compounded by hardware and software constraints. For example, we encountered individual tables for around 65 gigabytes, which is exceeds the available RAM in some environments. In addition, some secure platforms are not designed for multi-user access, or modern machine learning workflows. These constraints mean that the implementation choices are often derived not only by methodology, but also by what is computationally feasible in practice. And the main learnings here were the importance of incremental and database-driven processing. Taking these constraints into account early is critical to ensure that the OMOP pipelines remain scalable. And yeah, with that said, I end the presentation here, and I pass back to Vera. Thank you very much.
Thank you so much. Ioana and Claudia, how much time would you like me to take for presenting use case 1 results? I know we're almost at the end.
I think we can extend for more 7-10 minutes.
Okay, thank you. So I will continue presenting the use case 1 results, and as much as you have just heard from Mohammed about ETLing of the native data to the OMOP format, most of the results I will present will be in the native format, and I'll talk about it, about why. Also, we're working on a publication for use case 1 results that would include data in both formats, so stay tuned. So, let's see. Okay. So, as with ETLing data to a common data model format, there are surprises once you actually start looking at the data. So on the data access and analysis stage, we have planned to have a single analyst writing an analytical code for the beautifully OMOP converted data. However, we have encountered, I think in this project especially, severe delays to data access because of limitations, especially in Denmark, to with respect to data access from external partners. So we had to change data custodian mid-flight and to enable us to actually conduct the project as planned. But as a workaround to get some results ahead of the OMOPing of the native data, we actually went ahead and conducted all of the planned analysis in the native, so free OMOP format ahead of the analysis in the OMOP converted data. This had silver lining because of the challenges that Mohammed just mentioned. You don't really know when you apply the standard OMOP processes whether you're getting it right. So we, thanks to the data delay and the challenges, I should say, we had the benefit of having the native data analysis, format data analysis results as a benchmark for the results that we would obtain in the OMOP after the OMOP conversion. But the analysis themselves were relatively simple. We were interested in obtaining descriptive statistics of the breast cancer and ALS populations, including incidence rates, basic demographics, comorbidities, treatments, and some of the markers of disease progression and survival. So let me show you some of the high-level results. As I mentioned, a publication is in preparation. Let's start with breast cancer. So we have, along the way, as we as we obtain the results, always benchmark them also against the published evidence, although, as I mentioned, these results are not meant to be used in clinical practice. So the main message here in the graph on the left, you can see that breast cancer is primarily a female disease. You can almost not see the incidence rate in men, which is a very small blue rectangle. On the right-hand side, you can see trends in incidence of the breast cancer over this study period. Data are available for this patient population from Denmark and from Finland. They're not available from Portugal because of some of the problems that were identified on the data access stage. So the Portuguese team decided that would not be prudent to present these incidence rates data from Portugal. Also, I need to say that we are still waiting for data access in Germany, although this was planned as a 4-country analysis. We have data from 3 countries, and that also represents a real-world challenge in data access for regulatory purposes. So here are some of the results on the demographic characteristics of breast cancer patients. I would like to highlight some of the differences in the types of data available from the 3 different countries from which data were available. So for this specific data extraction, data on employment, for example, were available from Finland, were not available from Denmark or from Portugal. In Denmark, data on employment or other socioeconomic indicators would technically be available, but it would not be possible to have them accessed by third-party partners with the OMOP ready scripts. So here are some of the comorbidities that we have analyzed. We looked at the prevalence of psychiatric disorders, dementia, and Alzheimer's specifically, and cardiovascular diseases. You can see there is a lot of concordance, with the exception perhaps of cardiovascular diseases in Finland. We think that it has to do with completeness of recording, maybe with some definitions. We're looking a little bit closely into that. Most of the results are presented for women because, as you have seen, this is a predominant group of these patients. When we look at treatments 12 months post-diagnosis, you can see that there is some heterogeneity in the prevalence of treatment modalities such as radiotherapy, chemotherapy, and total or partial lymphadenectomy, but we see that for surgical procedures highlighted here, there is some relatively good concordance in reporting of the prevalence of these, and we think that this reflects generally a better recording of surgical procedures in the real-world data. When we look at the entire follow-up, we have up to 20 years of follow-up for some of these patients. We can see here some of the disease characteristics, and I would like to bring attention to heterogeneity of the way, for example, the tumor characteristics are reported in the 3 countries whose data are being analyzed. So, for example, Denmark and Portugal use the standard TNM staging diagnosis for breast cancer, while in Finland, an internal staging classification is used. And then you can see also that the distribution of the tumor size and the prevalence of metastasis at diagnosis, they vary quite widely across the data sources, and this probably represents for the most part differences in the data flow and completeness. When we look at signals of disease progression, we can see here that despite the heterogeneity of the data sources, some things are recorded pretty similarly and probably equally well. We know that malignancy diagnoses usually come from cancer registries, and there is not much measurement error, or as we say, misclassification in recording death data. So we can see that here reflected in the relatively good concordance across the data sources in these in these disease progression markers. And then you can see here, when we look at overall survival of breast cancer in the 3 countries, the graphs look pretty similar. So very quickly, we can see that men represent a much smaller group than women, men with breast cancer. And you can also see we observe in our real-world data analysis that men are tend to be substantially higher at diagnosis than women with breast cancer, which has something to do also with potentially poor prognosis in this subgroup. Okay, let me go quickly to the high-level results for ALS. Here you can see incidence rates from all 3 countries for which we conducted the analysis: Denmark, Finland, and Portugal. On the left, you can see that the data do reflect what we know from literature: slight preponderance of the male population in the ALS patients. You can see that the scale for incidence rate is much smaller than it was for breast cancer. On the right-hand side, you can see trends over time. You can see them to be maybe more heterogeneous than you think because of the vertical scale being pretty stretched out, but there are some differences. Also, you see here the populations of ALS are much smaller than the breast cancer populations. But you can see that there is good concordance in the demographics. You can see in all countries the median age and the socioeconomic, the median age is concordant, and you can see that in the country with the employment status available, most of the a majority of the patients are in the older age group and are pensioners. With respect to clinical characteristics of ALS, I would like to call out the heterogeneity of the data sources with respect to capturing things that are seen in this table. For example, you can see a large heterogeneity of other mild motor impairment. We also think that has to do with how data flows and what is actually recorded in the databases. When we look at treatment of ALS, 12 months post-diagnosis, as I mentioned before, riluzole is the only symptom-relieving treatment. This is also important to our plans for use case 2 in this work package. First of all, I would like to say that when we look at the things like intensive care and non-invasive ventilation, you can see differences in how countries report these. As you can see for riluzole, you can see that riluzole treatment is not reflected in the data in Portugal, although we know that riluzole is used in Portugal. And then the prevalence of use is higher in Denmark than in Finland, which might reflect differences in the populations, but also probably reflects some of the differences in the data completeness. So when we look at prognosis, we have a very good concordance in the risks of respiratory infections. And as we have seen in the breast cancer population, we have good concordance in the mortality data. And if you can remember in your mind's eye the way that overall survival graphs looked for breast cancer, you can see here that overall survival graphs looked much different for the ALS population. The drop in the first post-diagnosis years is quite steep. And we can see that across all the databases that we have analyzed. As I mentioned before, most of the analyses are available in native data format in all countries. However, we are able to compare, and we will compare in the publication, results obtained from native and from OMOP analysis. And this is just a teaser for you here. And the first thing, as an epidemiologist, you look at is the size of the population similar from the different sets of analysis. Here you can see data from Denmark and Portugal, the countries for which both sets of analysis are currently available. And you can see here that the sizes of your population, the concordance is pretty good. and so is actually the rest of the characteristics. So this seems to be reassuring for the future analysis. So to summarize the main findings and the lessons learned from this exercise, so as we have known, as we have learned again and again, data access delays is an important barrier to timely generation of real-world evidence, and correctness of data delivery cannot be automatically assumed and needs to be subject of standard quality control procedures for researchers before they start using the data. The differences, the heterogeneity of the results across the country-specific patient populations stems from various sources. It can stem from true differences, but also from artifacts of data flow, from the completeness of recording, from things like reimbursement practices, and from the data provenance. Are we talking about administrative data? Are we talking about claims data? That all matters. The heterogeneity of the data, including data type, validity, and completeness, should be used for assessing how fit for purpose the data are in relation to specific research and regulatory questions. And depending on the research question, the answer could be different for different data source. So in conclusion, we say that use of real-world data in regulatory pre-authorization decisions requires expert knowledge to assess the impact of data limitations. Harmonization of data to the same structure does not remove underlying heterogeneity of data quality, flow, and completeness. And I just want to stress that the results for breast cancer and ALS are generated for illustration purposes only. They should not be taken as indicative of disease epidemiology. However, they are broadly consistent with the published evidence. So thank you so much for your attention. I hope we have a nice discussion following this.
Okay, thank you very much, Vera and Mohammed. And I will pass over to Britta for the roundtable discussion. Thank you.
Yeah, thank you very much, Claudia. And also thank you very much from my side to Vera and Mohammed for this very exciting presentation. And now it's time for our roundtable discussion. First of all, I would like to introduce the discussants. We have Pia Rannenheimer. Pia is the Chief Specialist at the Finnish Medicines Agency and the Development and Information Services. Her primary responsibility is to lead the development of Finland's National Medicines Data Repository and its online services. Pia has over 10 years of experience in HTA and worked in Fimea's HTA team until 2022. She currently serves on the Finnish Council for Choices in Healthcare, PALCO, as a council member and vice chair. PALCO issues national recommendations on whether hospital-only medicines should be included on the publicly funded healthcare service basket in Finland. So very warm welcome to Pia. Then the next discussant is Lucia Ortiz. Lucia Ortiz-Lo is a health biologist and virologist. She currently works as a research and communication project technician at the Spanish platform of patient organizations. She is involved in the world of associations after being diagnosed with congenital heart disease. After operation and successful recovery, she realized that no one knows the reality of being sick better than the patients themselves, and that their experience should be taken into account in all health-related decisions. Also, very warm welcome to Lucia. The next discussant is Daniel Morales. Daniel is a senior clinical epidemiologist within the Data Analytics and Methods Task Force at the EMA, also working closely with Darwin EU, and his background is in general practice and academia. Well, welcome to you, Daniel. And the last discussant is Ruben van Eyck. Ruben is an associate professor and biostatistician at UMC Utrecht, the Netherlands. He previously served as a visiting scholar at Stanford University's Center for Innovative Study Design. His research focuses on integrating real-world data into clinical trials and developing new endpoints that address both multidimensionality and also patient preferences. Also, a warm welcome to you, Ruben. So now we have prepared some questions, and I would like to ask the discussants to answer the respective question from your perspective, from your point of view, and please taking no longer than about 5 minutes for your answer. So the first question, so we heard the presentation from Vera and Mohammed, and during the presentation, there were some barriers also to real-world evidence use in the pre-authorization stage. Mentioned, for example, the lack of awareness about the data sources that are available, or concerns regarding data quality, or also delays in data access. So in your opinion, what contribution can the stakeholder group you represent and belong to provide to overcome these obstacles? As first, please, Pia.
Thank you. So do you hear me well?
Yes, we do.
Perfect. So I have experience in one prior OMOP-based project where we piloted the data from Finnish OMOP centers in 3 different use cases that concerned multiple myeloma, SMA disease, and so on. And I think we encountered the same issues that you see here, like with the time delays and so on and so on. So for me, as someone who represents HTA and decision making, I think it's very important for us to describe the evidence needs that we have to, groups like this that run the analysis, but also to the OMOP centers and the national institutes that sort of steer the data management and the processes development nationally. So for me, I think it's important that we as HTA personnel really describe our evidence needs thoroughly so that the systems can develop to answer our needs. So for example, here I see that the breast cancer and the ALS examples are covered in really high level. And when we make decisions on new medicines, we always look at the specific indications that these products are intended for. So this is a really strict demand also for the data and evidence that is produced, that we would like to see sort of patient group specific results. I think that's my first reflections.
Okay, thank you, Pia. Then it's Lucia.
Hello. Can you hear me?
Yeah, we hear you well.
Okay. Well, thank you for inviting me and for having us patients here in the conversation. I won't be long answering this question. I think that from the patient organization side, our mainly to increase trust, usability, and speed of the real-world evidence in pre-authorization. And how can we do this? Well, we do this by improving awareness of data sources, because we patient organizations usually act as connectors between the participants, which are the patients, and the regulatory bodies. And also we help them to translate technical documents into plain language so that they can understand every step. And we can also help them by focusing on variants and data that are actually relevant for them. And we can help them define which endpoints or outcomes are meaningful in their real life to make some regulatory decisions.
Okay, thank you, Lucia. So now it's Daniel.
Yeah, thank you very much for the invitation, and it's great to see all the work being presented as well. I think in terms of the elements in question 1, I think in terms of a greater awareness of data sources that are out there, then from an EMA or European Regulatory Medicines Network, we have the HMA-EMA catalog of data sources, where data sources, descriptions and metadata can be added. And eventually, through the European health data space, there will be a larger sort of data catalogue anticipated covering other uses than just medicines regulation. In terms of the quality, I would suggest that the EMA data quality framework was published at the end of 2024, and there's an extension to the data quality framework that's under review specifically for real-world data, because there are nuances specifically with real-world data, and we hope then that will improve both the data metrics, their transparency, and the reporting that can then hopefully help assessments as well. I think another aspect that the European Medicines Regulatory Network can help is providing scientific advice. So if there are uncertainties about its use and its relevance of real-world data for a specific question, then applicants can seek scientific advice before either conducting the study or submitting. But equally, data sets and data sources that are maturing can also seek qualification. And that is also a process that can be viewed. And I think delays in accessing, I think we've all faced that, anybody who's tried to do projects with real world data, and that's a really difficult thing to address. So within the Darwin EU network, in some instances, but not all, we've been successful in trying to use blanket protocols that tend to be for simpler study designs, and some IRB committees have accepted those. But we hope then something like the European Health Data Space will also facilitate that with standardized applications and maybe stricter timelines for when maybe approvals or data can be provided. So we hope that that landscape will support as well. Thank you.
Thank you, Daniel. So now it's Ruben.
Yeah, thanks. So I think I agree with what Moses already said. So yeah, from my perspective, so more the academic perspective, I think like the data access issue, yeah, that's the highest priority, and you saw it also in your project that is causing like a main delay. So the project that we have ongoing, like where we try to use real world data, for example, in clinical trials, or where an industry partner tries to require data from an academic institution or from one of the national registries, yeah, it sometimes becomes almost infeasible just because like access to patient level data, that's very difficult. And I think that's maybe where the most effort is needed. So how can we make this happen? And so I speak mostly from the ALS perspective, but what we have done there, for example, is that we as a community have created like a common data set, so where we already try to use something similar as the OPM system, or the OMAP system, where you try to standardize like all of the fields and the outcomes, and try to merge that and make that open access to the community. So that's called PROACT at the moment. So that consists out of registries and consists out of trials, and basically the whole community is contributing. So both the academic institutes and like the investigator-initiated trials, the registries are contributing, but also the industry are contributing as well. And that I think creates a very nice system. So industry itself is contributing, but can also access these data then for their own development. So that is an important point, and I think the detail of data that's often required for these projects requires quite a lot of detail. So if I reflect a bit on the results that were presented, like for ALS, like very important prognosticators like quality of life or like actual disease status, these are usually not really captured in electronic healthcare records, but these are really the outcomes that we're looking at in drug development, and also in order to make prognosis. So I think that there is some effort needed as well. So how can we collect like these more difficult outcomes? And so one of the ways that we're setting up now in Europe is to create like a common registry, so where each country tries to collect like in a harmonized manner a core outcome set. So where we collect like quality of life and functional measures of patients, but also like the basic diagnostic information. So how long is a patient sick, and details about the diagnosis. So these data are not standard captured in electronic healthcare records, but yeah, so that has to come really from the community to make such a registry, I think. And yeah, so I will stop there for now.
Thank you, Ruben. So a lot of input to this question with different perspectives concerning the data sources, trust in data sources, data access, all these things are, of course, difficult to overcome the problems in most cases, but there are some initiatives, so that's very good. So the other topic that we had in the presentation was about OMOP, and I would like to ask you, so what problems does OMOP solve that the well-designed local data model does not? So concerning native data analysis, so what does OMOP solve, and what are the advantages of OMOP or other common data models there? So we again start with Pia.
Thank you. So I think the basic principle or aim to have like semantic interoperability across borders, that's the key issue if that's accomplished. And so to be able to combine data sets and so on. I think in federated analytics, the basic principle also that the data stays local and then the analysis can be run separately. That's also something that may speed things and create more trust to the system. One thing that was on my mind was that we don't have many national disease-specific registers in Finland, and I think that's one issue what the OMOP standardization could do, that we could combine data from, for example, university hospitals more easily. And when we do have quality registers, for example, the level of detail in those registers is not usually so deep that we are on that like precision medicine stage where we know all the details about the disease or the patient groups that we would like to have for fit for purpose decision relevant analysis. Thank you.
Thank you, Pia. Then it's Lucia.
Thank you. Well, I agree with what Pia just said, but I don't think that OMOP doesn't maybe compete with a good local model in how well it analyzes, but it solves the problem that the local model has. For example, problems such as scalability and comparability that happens across organizations and countries. And as an example of an advantage of the OMOP model, I would like to, if you don't mind, talk you about one of our tools that was standardized by, well, using the OMOP model, and it's called Groby. Groby is the first validated in scale that allows us to understand the impact of the disease in the psychological, affective, emotional, and social aspects of the chronic patient's life. So in this way, thanks and because of using the OMOP model, we can use the results of this scale to highlight the invisible aspects of people with chronic diseases and use them in regulatory health.
Thank you, Lucia. So then it's Daniel.
Similar to other colleagues, I think one of the benefits is interoperability that allows once it's set up, and there's obviously quite a high upfront cost in preparing and transforming data to a more common data model, especially if it's for creating an ETL for the first time. But once established, it certainly can facilitate or increase the speed of federated analytics that can be performed, and also scalability. Also, there's governance benefits for cross-border analyses. And then the standardized analytics can reduce one aspect of heterogeneity, which, and maybe if running on your local data, there's some local assumptions required for protocol interpretation. Thank you.
Right, thanks.
Yeah,
And Ruben.
yeah, I agree with everything that's said. So I think like the yeah, like the main use of real world data, especially in a rare disease, is like international exchange of data sets, and there like a common data model really helps. I see also a risk of using it because it seems to be very time consuming, and yeah, like really to set it up. And so I was wondering if like some aspects maybe you should prioritize, like, and others not. So yeah, maybe it's more like a hybrid form I can see. So like a full model might sometimes be like an overkill for a project, but it depends also, I think, on the ambitions.
Yes, indeed. So there are, of course, advantages with using OMOP, but there are also, of course, advantages using native data or embrace also heterogeneity of different data sources. We also presented some results of our use case one, and concerning ALS and breast cancer. And our question would be, did you find an implausible finding for ALS or breast cancer? And then can you explain why it is implausible and speculate on the reasons for this? So this is also something that's maybe worth discussing. Pia.
Well, I think my attention was on the question like, was this really comprehensive for Finland? Was the dataset comprehensive for Finland? Because I know that we have 3 OMOP data centers that are university hospitals. So I think to assess what's implausible or not, like more background information on the methods would have been needed. And as I said in my first response, I think like we always look at really indication specific subgroup from subgroups these topics. So I think for me, reflecting to those current like decision maker needs, this was on a like too broad level, the results.
Okay, thank you, Pia. Lucia?
Yeah, well, thank you for the question. Here at POP, our organization, we don't really discuss about specific disease because we represent patients with chronic conditions. So I don't really have to say much about ALS and breast cancer specifically. But I think that those implausible results data mapping issues and spotting them early, I think it could help to avoid wrong conclusions in regulatory or HTA decisions through the process. So thank you.
Thank you. Daniel?
I think the implausible ones were slightly highlighted during the presentation, and I think appropriately so, recognizing this likely a data capture aspect. For example, I know the weather's much better in Portugal, and that helps. But in the breast cancer analysis, there were nobody with a past history of psychiatric disorder, for example. So, but I think that's sort of the importance of understanding the data ahead of time, characterizing the data and generating data metrics. But also importantly, I think it requires for some of the things an understanding of the clinical practice guidelines within that country as well, to really know. So I don't think it could always be an assumption of data quality. I think they should go hand in hand, and that's sometimes a really challenging aspect, especially with federated analytics and the heterogeneity that we have across Europe. So.
Indeed. Ruben?
Yeah, so I was mostly reflecting on the ALS data. So there, yeah, I noticed like a few discrepancies like within the care use. So for example, Riluzole use or like some kind of medication use that seems to be very different across countries or a little bit low. So for example, the 30% Riluzole use, like the main medication use for ALS, seems off. Well, we know that like around 70-80% of the people are using it. But yeah, it could be like a true care practice difference across Europe. So it would be good somehow to position these data to like population-based registries, and what do we know from there? So how well do these results match with what is published already in the literature? Again, like what I said in the beginning, is that actually you're interested in much more detailed disease characteristics. So it could be like how severely affected were these patients and how long were they sick, and this is very important to know clinically in order to know whether these data make actually sense. So for the oncology, so the breast cancer case, you had like the staging severity. So it would be good to have something similar for ALS, so that makes it easier to interpret the data in the end.
Indeed, and also heterogeneity in results may also depend on recording and reimbursement practices, data provenance, so that all that you mentioned, really. Thank you. So one of our last questions is, so given the methodology used and also the results obtained so far in Real4Reg, how do you think a project like Real4Reg can be helpful in regulatory decision-making, or also for a specific real-world data, real-world evidence research questions, and which impact will it have in your work? Pia.
Well, I'm speaking from the HDA and national steering perspective. So I think real-world data is self-evident that it's important in all stages of medicine's life cycle, and for us as HDA bodies, we have a real task sort of to describe what are our evidence needs. For projects like this, like every decision maker group, every steering group has a little different evidence needs. So I think it's also relevant to really specify which sort of customer group are you aiming to serve with your data products and data services, so that you can customize. So regulators have we have similar data and evidence needs, but there are differences in HDA perspective and so on and so on. So I think that's crucial. And then also we know that the OMOP sort of data basis needs development, and then also the processes where we sort of ask for data and you produce evidence for us, they need development. So perhaps these should be done in separate projects, like developing the local data lakes and data warehouses and data management, and then having these international services for accessing data, analyzing things jointly, and so on and so on.
Yes, thank you, Pia. Lucia?
Yeah, well, as patient organizations, we think this project is useful, mostly because it can provide a stronger evidence for patient advocacy in order to making regulatory decisions. More outcomes that are actually relevant for patients in the conversation. So for us, the impact of this project is really practical, because I think it can make real-world evidence more reliable and explainable. So then it can be used in decision that affects patient access and quality of life. So thank you.
Thanks, Lucia. Daniel?
These projects are very important. There's a temptation sometimes to go straight for the clinical information and evidence generation, but understanding all the processes to get there are really important for trust and transparency. I think if we stick with the use cases of breast cancer and ALS, for example, there's certainly great potential to establish a set of data metrics that could be run against the different databases for such real-world evidence studies of breast cancer and ALS, certainly to determine fitness for purpose, but also to identify data gaps that over time, because databases have a life cycle, may also be enriched through different means, either through free text AI or something like that in future. So I think that's a really good potential there. I think the other aspect is the challenging around OMOP. I have to remember that OMOP had a very traditional US evolution, and we almost need to Europeify it, if that's such a word. Okay, and that's kind of new ground. So I think all of those learnings, there's a temptation that everybody's doing this for the first time, and that might itself introduce a bit of heterogeneity. So I think being transparent in how you devise that sort of OMOP mapping, try to get that out publicly and accept and adopt it for certain areas. Some things are very cancer specific, others are certain disease specific, even within cancer, then I think that can only help the landscape over time as well, with any recommendations for the learnings you have.
Indeed. Thanks, Daniel. Ruben?
Yeah, no, I completely agree with what Daniel was mentioning. I think I have very little to add. So I like to see this project also like as a benchmarking data set. So how, like, from an epidemiological perspective, like how the disease is, like the actual numbers on the disease, and like how prevalent is it, what are the differences between countries. So that could be very, very valuable, I think, and have a lot of impact. But yeah, I think like the exercise itself, that's maybe the most valuable. And also to learn like what are the complications there, and maybe as a field, how can we adjust things so that we can capture like missing aspects now, and like how would we like to progress. So I think it's an extremely important project. Thank you.
Yes, thank you, Ruben. So as the workshop is near its end, now it's time to summarize. So let me shortly sum up. So today we heard about the first results of the use case 1 on Real4Reg and the implications of the findings. So the use of real-world data and regulatory pre-approval decisions requires, of course, expert knowledge, especially when it comes to impact on the interpretation of also the data limitations. And also when we have a look at the regulatory side, of course, evidence needs need to be adequately addressed with real-world data for generating real-world evidence. And it's also very important to be aware of the strengths and limitations of the data sources and the data quality, and to conduct standard quality control procedures before the use of data. So that also the trust in the data sources can be increased also from the patient side, for example, so that everyone is aware that the data sources are used also scientifically relevant and in the correct way. And sometimes it can also be time consuming to reach the right data source and to receive the variables you need, as the data access can be quite a bottleneck, as we all know from the different projects. And it can really be a kind of a barrier to timely real-world evidence generation. And it's a recurrent challenge across countries and a lot of data sources. And of course, this needs to be addressed probably in more detail for the future. We also heard about the OMOP process. So the harmonization of the data to some structure does not remove, of course, the underlying heterogeneity of the data quality or flow or completeness. And while common data models like OMOP are very useful in interpreting results from different backgrounds and countries, it's still necessary to embrace also the heterogeneity of the native data, especially in view of different reimbursement practices in different countries, different recordings, or the different data provenance from claims data to registry-based data and so on. Of course, there are also some initiatives also for better data access, and also a catalog of different data sources, especially the EHDS, European Health Data, where we'll have large data catalog available. And yeah, so the heterogeneity should also be used for assessing a given data source and its fitness for purpose in relation to the specific regulatory but also research question concerning real-world data and real-world evidence. Concerning the regulatory side, it's also important to have a look at the scientific advice also, because it can also help to get the suitable data sources involved, especially when you think of industry-related research or market authorization processes. Okay, that's it for today. So now I would like to thank all the presenters, roundtable discussants, and all the workshop participants, of course. And I would also like to give a big thank you to the INFARMED team for organizing the workshop, and a big thank you all to all Real4Reg partners who contributed to the generation of the presented results today. So thank you very much again, and have a nice afternoon. Goodbye.